The human tau protein is encoded on chromosome 17, position 17q21, and contains 16 exons. The protein occurs mainly in the central nervous system and consists largely of 6 isoforms generated by alternative splicing of its mRNA. The 6 tau isoforms differ from each other by the presence or absence of 1 or 2 portions, containing 29 or 58 amino acids in the N-terminal part and by the presence of either 3 or 4 repeats in the C-terminal half. The N-terminal inserts are highly acidic. The repeats in the C-terminal half of tau are the domains by which tau binds to microtubules. The tau proteins found in nerve cells, particularly being active in the distal portions of axons, where they stabilize microtubules and provide flexibility. The tau proteins provide the stability through isoforms and phosphorylation. However, when the tau protein is hyperphosphorylated, it loses the ability to bind to the microtubule, causing the microtubule to unwind, making the once healthy neuron diseased. This hyperphosphorylation of tau alters the helical and straight filaments of the protein, causing it to tangle, creating what we know as neurofibrillary tangles, NFT, or tau tangles. These tau tangles have been hypothesized to contribute to the pathology of Alzheimer's disease. The phosphorylation level of tau autopsied from an Alzheimer's disease brain is 3 to 4-fold higher than that of a normal human brain. The tau tangles clump together very tightly, making an insoluble form. The precise reason of why tau can hyperphosphorylate and tangle is not completely understood. However, when it is solved, a cure for Alzheimer's disease is just around the corner.
Alzheimer's is defined as a neurodegenerative disease characterized by the progressive loss of memory, cognitive thinking, and behavior. In the early stages of Alzheimer's, people may experience irritability, anxiety, and depression. In advanced stages of Alzheimer's, there may be anger, verbal or physical outbursts, restlessness, sleep disturbances, and more. While we do not know for certain, scientists hypothesize that the tau tangles, along with another protein buildup, beta-amyloid plaques, are the direct cause of this cruel disease. The beta-amyloid protein is normally found in the human body. However, for an unknown reason, the protein builds up in the space between nerve cells, called the synapse. This buildup causes the interruption of communication between nerve cells. This form of the protein is known as beta-amyloid plaques. As a pair, the tau proteins and the beta-amyloid plaques are thought to accumulate inside the brain, somehow reacting with one another, causing healthy neurons to begin to work less efficiently. These once healthy neurons begin losing their ability to function and communicate properly, eventually causing them to die. This causes the brain's tissues to irreversibly shrink. While there is no cure for Alzheimer's disease, some medications are commonly used to help ease behavioral and psychiatric problems. Attempts to find a cure for this life-stealing disease have been unsuccessful. However, with the advancement in the research of the tau protein and how it tangles, proves that we are persistent and hopeful to find a cure.